Hey everybody, today we're going to talk about the modes of inheritance. Now there's quite a bit of a list here. It's important to understand that there's some overlap with some of these and that these are just ways for us to sort of break down and understand some of the key differences in how different traits and characteristics are inherited. All of this stems from the work done by Gregor Mendel, and really has grown from there as we've gained a better understanding of the actual concept of DNA and the genetic material, and how chromosomes are moved around and shuffled and inherited. So we're going to start off with a chat about dominant versus recessive, where I think we might kind of redefine those terms compared to how you've previously learned about them. But if you look at this list, you probably fall out around the second or third one in terms of things you've talked about before. But no need to feel overwhelmed. Like I said, each of these is just sort of trying to tackle a specific scenario of difference in how things appear, even though they all kind of stem from the same general idea. So, recessive and dominant. As you traditionally learn this, we have dominant genes that are the ones that show, and we have recessive genes that are the ones that don't show unless you have two copies of the gene. That's often how it's taught, and it's not necessarily incorrect, but it kind of misses a key point about what's really going on here. And that kind of shows itself when you start to think about heterozygous individuals, and you wonder, well, why does a heterozygous person, a person who has one dominant and one recessive allele for a particular trait, why do they only show the dominant? Like, they have the recessive, so why don't they show it? And we kind of explain that away when we're not diving too deeply into this by saying, oh, well, the dominant one is dominant. It's the one that overpowers the recessive trait. And again, that's not wrong, but I think it gives the wrong idea. And the reason for that is kind of simple. Dominant genes are functional genes, and that means that they code for proteins that actually do a thing. And what that means is that a recessive gene is one that doesn't code for a functional protein. It doesn't produce a protein that does anything. And so when we talk about recessive traits, we're not talking about, in the case of no cleft chin, you have the no cleft chin gene, and so it makes you not have a cleft chin. Well, if we think about it, it makes a lot more sense that, well, if you have the cleft gene, then you show that trait because it produces a protein that, for whatever reason, in the development of your chin, produces the cleft chin. If you are recessive for that trait, you just don't have a gene that codes for a protein that makes you have a cleft chin. You have a non-functional protein, and so your chin is just whatever it already was. And if we look at any dominant recessive alleles, that's what we find, is that the dominant one is producing an effect and the recessive one is just failing to produce that effect. So if we think about like eye color, and traditionally, we oversimplify eye color a lot, but if we take that traditional, brown eyes are dominant and blue eyes are recessive, it's not that you have the brown eye gene, the brown eye allele, and it gives you brown eyes, and you have the blue eye allele and it gives you blue eyes. Rather, you either have the brown eye and it produces pigment that gives you brown eyes, or you don't have the brown eye allele and your eyes are just still blue because they already were because of some other gene or some other influencing factor that made them that color by default. So it can often be helpful to think about recessive traits as being just you failed to change it, and so you're sticking with what it already was. And of course, that gets complicated. Eye color's, one that really shows you how complicated it is, because eye color, in truth, is governed by multiple genes, lots of different, development of pigments and things. But it's important to really understand this idea that recessive alleles are ones that don't have an effect. They may still code for a protein. They likely do. They may still get expressed. Those proteins get produced, but those proteins don't do anything. And so that's why, if you look down this list, these are really good examples to help you to understand, because in each one, it's kind of like you have the gene that does the thing, or you don't have the thing because you don't have an allele that causes it. Going back to heterozygous individuals, the reason why this makes sense is because if you are heterozygous for having a cleft chin, you have one copy of the allele that produces the proteins that gives you the cleft chin, so you have a cleft chin. And then you have another copy of the protein that doesn't produce, another copy of the allele that doesn't produce a protein that gives you a cleft chin. It doesn't matter. You still have one copy producing the proteins, and therefore you still have a cleft chin. And that's why we can't tell the difference in typical cases of dominance and recessive of individuals who are heterozygous versus individuals who are homozygous dominant, and they have two copies of that dominant allele. So there are two potential phenotypes. Either you display the trait because you have one or two copies of the allele that are dominant and effective, or you don't display the trait because you're recessive. One last point on this, this bottom part here. It is a wide misconception to think that, recessive is less common. It's going to be overshadowed by dominant. It's going to be, just a matter of time before it's gone from the population. Statistically and just otherwise, that's not true. In fact, looking down this list, we can see lots of examples of recessive traits that are far more common than the dominant other half. While it's true that in cases where things are being selected for the characteristic or the trait, then we will tend to see the dominant more and more because of heterozygous individuals showing the trait and such. In truth, the allele frequencies of these things are going to remain roughly steady unless they are being selected for, and that's a concept we'll get into with evolution, but just please know that there's nothing about a recessive trait that makes it less common. There are many examples of recessive traits that are far more common than the dominant version that we might have as an alternative. So another one that you've probably already heard about and feel pretty good about is incomplete dominance and codominance. These are pretty straightforward examples of just what happens when there is more than one dominant version of an allele. And so taking what we learned on the last slide and applying it here, we can understand that, well, if you have two different versions of a protein that both do a thing, and maybe it's a different thing, but they both do it, then if you had both copies of it, you would show both of the effects because you'd have two functional alleles coding for two functional proteins, and those functional proteins would express themselves. The only difference between incomplete dominance and codominance is just sort of what the phenotype ends up being. So in the case of incomplete dominance, we see sort of a blended effect. And so that example would be like the pink flowers here. So if you have an allele that codes for red and another version of that allele, another version of that gene that codes for white, then we would see pink. Because if you zoomed in on those flowers, what you would find is that they have both the proteins that cause the red pigment or red coloration, and you'd see proteins that cause the white coloration. And so when you zoom out, you see the effects of both of those blended together. That's incomplete dominance. And the name just sort of implies that neither one is really more dominant than the other. That gets confusing because codominance is kind of the same thing. It's just that in terms of what it actually ends up looking like can be notably different. And so we give them two different terms because in codominance, what you can find, and here's an example, and a lot of examples have to do with flowers, going all the way back to Mendel. Florists are genetic masterminds. I think we should give them a lot more scientific credit than we do because what they're doing is blending and mixing between different individuals in order to produce sort of a final product that is a culmination of genetic interactions, which is pretty cool. But in this example, we see red and white, and we end up with a flower with petals, some that are red, some that are white. Now, this can be more complicated than we're really explaining here, and that's just because in some of those petals, there might be other factors that might lead to the expression of only the red or only the white. And so in that way, it makes it a little bit different than incomplete dominance where, each of the petals is just showing both. And so you see the blended effect. Another great example that comes up a lot of codominance is blood type. And so with blood type, you have, let's just take the A, B, and O. You have two different functional versions of a protein, A and B. Those are the two different versions that you could have. And then you have a recessive third option, which remember means that it's just non-functional. It doesn't produce a functional protein, and we call that O-type blood. So a person who is AB, for instance, for blood type, would have two different alleles, both coding successfully for proteins that show up on the surface of their blood cells. And so if you looked at that person's blood cells, you would see both the A protein on its own doing its thing and the B protein on its own doing its thing. And so you would have both versions being present and visible. We would call that codominance. The next one, another one probably have discussed, is sex-linked. Now this is the first of three that are going to be very easily confused. So we'll want to take a second and really kind of understand those as they compare to each other. Sex-linked, these are genes that are located on the X or Y chromosomes. Most often the X chromosome, as it's a more substantial chromosome, size-wise and in number of genes. Now, the reason why this matters for individuals from an inheritance standpoint is because karyotypic females have two X chromosomes, whereas karyotypic males have one X chromosome and then a Y chromosome. Now, the Y chromosome does things, but largely we can consider it kind of non-functional for the sake of many of the different potential traits that it could code for. And so when we consider this idea of being sex-linked, it matters because a karyotypic male is only inheriting a single X chromosome. And so if they inherit one, let's call it bad copy of an allele on their X chromosome, they don't have another X chromosome to rely upon to produce the functional gene or to produce a better version or whatever. And so in that way, certain traits are easier for males to inherit because they only have one X chromosome. Color blindness being an example. In the case of a colorblind male, they have inherited their X chromosome from their mother, and that X chromosome contained some faulty allele for some color blindness gene, and there are many different types of color blindness because there's many different genes involved in the development of the different cells that produce our color vision. But in the case of color blindness, a male inherits a single copy of a non-functioning allele, and so now they have limited color vision, versus females who would need to inherit two faulty copies. So they would have to have a colorblind father because he only has one X chromosome to give, and so he would be colorblind himself, would pass on his colorblindness gene to his daughter, and then they would also have to have a mother who is at least a carrier for colorblindness, and then have the bad luck of getting that gene from her. So in that case, it is much less common for karyotypic females to exhibit color blindness. Other examples are hemophilia, ichthyosis. You can find lots of examples of all these things. I've only chosen ones that are kind of memorable and interesting to talk about, but you can find lots of examples of all these things. So that's sex-linked. Now the next one is sex-influenced. Now these traits are autosomal, which means they're not necessarily on X chromosome or Y chromosome, so they could be on just any of the other chromosomes. However, their expression is going to be different depending on whether you are karyotypically male or female. And the reason often comes down to hormones. And so the presence of, for instance, testosterone produces the different effects of pattern baldness. So pattern baldness is a trait that can be inherited from your parents. However, if you're male or female, it expresses differently, simply because the expression of that gene, the actual functioning of the proteins that lead to the hair loss, is going to be influenced by the presence of other factors that have to do with your sex. So in the case of pattern baldness, the presence of testosterone means that these proteins do whatever their thing is differently than it would in a person who has much less testosterone. So that's sex influence. So they're still expressed, but they're expressed differently. The third one is sex limited, and this is where we only see expression in one sex. And so in this case, these are, again, autosomal, so they're not on the X or Y chromosome, but these are only going to be expressed by males or females. Again, for a variety of reasons that are bigger than this or outside of this, other genes, other factors. It could also be hormonally regulated, but the key difference between sex limited and sex influenced is that sex influenced is expressed in both just differently, and sex limited is only expressed in one. And so I included the example here of coloration in birds. So male and female birds, males are often much more pronounced colors, and they're the ones that have to do the whole song and dance in order to find a mate. The expression of the different, for instance, pigments in this guy here they both may have the genes for that, but he's the only one who actually successfully expresses those genes. So there are other factors, transcription factors or something along those lines, that are only activated in him because he's male and are not activated in this bird here because she's female, and therefore doesn't produce the effects of that at all. So it's not that it's expressed in both here, it's only being expressed by this guy. Another example is milk production in mammals. So both male and female mammals have all the genes, the genetics for the production of milk. However, in females it happens because of, again, other factors, and in males it is not expressed. All right, so actually let me take a second. So sex-linked is on the X or Y chromosome. Sex-influenced is expressed in both. It's not on the X and Y chromosome. It's expressed in both, just differently. And sex-limited is it's expressed only in one or the other. Cool. X inactivation. So kind of on the same wavelength as the last few, this one comes down to sex chromosomes. So having two X chromosomes working in the cell is actually kind of problematic. So because karyotypic males have an X and a Y, and the Y kind of just doesn't do a whole bunch, the animals with this situation have evolved to really only need one of these chromosomes. So the sex chromosomes really are separate from the other chromosomes in terms of how they operate, because we only want to have one single copy in operation at a time. This goes deeper, and it goes into the territories of chemistry, as the number of chromosomes that you have influences the rate at which different proteins are being expressed, which changes are being transcribed, which changes the concentration of those proteins, which influences their action within the cell, and all sorts of things. And so these organisms have evolved to only need one copy of this in order to have the right amounts of things. So what that means is, in a female organism that has two copies of the X chromosome, one of those needs to basically be shut down. And so it kind of gets crumpled up into something called a bar body, which I like to think of as a wad of chromosomes. So it just kind of gets like packaged up, pushed to the side, and it's not going to do anything. And that's no big deal because there's two X chromosomes. And so who cares? We crumple one up, we put it to the side. This will happen on a cell-by-cell basis, and so it's a permanent thing. So once one is sort of chosen, and that can be random, there's maybe influencing factors. I don't know enough about that to really speak to it, but think of it as basically just like, we picked this one for this cell, and here it goes. And so what that means is that X chromosome now is the one that's going to be expressed and is going to produce proteins that do a thing. In humans, there are traits that are related to which X chromosome you happen to be expressing, but there's nothing that's like overtly physical. So you can't look at a person and look at their skin and be like, oh, it's X chromosome number 1 or it's X chromosome number 2. You can't see that difference, but you can in cats, because the fur color gene happens to be on the X chromosome. And so the interesting kind of example here is calico cats, which every calico cat you've seen is almost certainly female. And the reason for that is because the coloration, the patterning of the light and dark fur color that happens in calico cats happens because one of their X chromosomes got crumpled up in early stages of their development, and the other one is expressing the color. And then on some other section of their body, it just happened, random happenstance, that this X chromosome was the one that ended up sticking around, and this one got crumpled up, and so they show this other color over here, and then that's happening in all these little pockets all throughout their skin, and so that produces then the different hair colors that you see, and so that patterning is only possible because they have two X chromosomes, which is why you're not going to find any male calico cats. Those are just orange or brown cats. They are genetically capable of being the same sort of pattern. Sorry, I misspoke. They're not, because they only have one X chromosome. They are the same kind of cat, and then they reproduce with a female cat, and they could produce kittens that are calico or whatever. But the point is that by being male and only having one X chromosome, they only have one color to show, and so they just show that color. Whereas females, there's an option of which X chromosome happens to be expressed. Tortoiseshell cats are the same way. But like I said, in humans, there's no like overtly physical characteristics that happen to be on the X chromosome, so you don't see any specific patterning or things like that because of it. Polygenic, this is easy. This is just the fact that it's multiple genes that cause the final phenotype. So skin color, hair color, eye color, all good examples, easy to understand examples of polygenic inheritance, because in these cases, like in the diagram here, for skin color, there's primarily 3 different genes that are largely responsible for skin color, although there are also more that can be. But what that means is that there's a gradation to sort of what your skin color could be because of the dominance or recessive or heterozygous nature of a whole bunch of different genes, and so then it produces in the end a final product. So anytime you see sort of variety in a particular physical characteristic, you can be reasonably sure that it's a polygenic thing, because if it was just dominant recessive, there would only be 2 options. You have dimples, you don't have dimples. That might be a bad example, because I imagine there's different degrees of having dimples, so that probably also fits in the category of polygenic. All of this stuff, you have to remember, is probably more complicated than we ever give it credit for, just because we have a lot of DNA. Other traits with complexity, facial or bone structure, height or weight, the genetic influence on those things can be massively varied, and so that tells us that there's most likely multiple genes that together are going to lead to sort of the final product. Pleiotropy, this is a cool one. So in this case, this is I say it's a cool one. It's actually often linked to all sorts of genetic disorders that are terrible, but this chicken's kind of cool. So in this case, what we're seeing is a gene that has an effect, but then it also has like some other effects that happen to like have nothing to do with the first effect that we pointed to. And so it shouldn't really be too surprising. We have such a focus of, the craziness of chemistry and stuff inside of these cells. In the case of this chicken, for instance, it has a trait that codes for a protein that gives it these curly feathers, which is atypical for most chickens. So it has some curly feathers. Well, it's also been discovered that the curled feather chickens also have kind of weird body temperatures and more metabolism and higher blood flow rates and greater digestive capacity, whatever that means. So the point is that the presence of that protein, it's a protein inside of its cells, and proteins do things. At the very least, proteins in their presence can influence the activity of other proteins nearby just by virtue of their concentration or whatever. And so stuff's complicated is what this one comes down to. The fact that, oh, hey, we've got this protein, it does a thing, cool. Oh, whoops, unintended consequence, it also does these other 12 things or influences these other 12 things inside of the body. That's what pleiotropy is all about. Sickle cell anemia is another example where the single little protein change in a hemoglobin can lead to fundamental shape change of blood cells, which can be massively problematic, but can also have a trickle-down effect of like a million different things in the human body. Just so we point to that as pleiotropic effects, that this is a singular change in a gene, but it can have this varied and just myriad effects. And the last one, epistasis, more cute animals for you. My favorite example of this is in labs. lab dogs, Labrador dogs. And the fact that if you look at a chocolate lab versus a black lab versus a yellow lab, they're all the same. They're all the same, but you get these varied colorations, and it comes down to two genes, which is neat. So epistasis is where you have the expression of one gene directly linked to the expression of another. And so to explain that, this dog right here on the left, this yellow lab, is actually a chocolate lab or a black lab. We don't know because there's a separate gene that shut down the production of the pigment. So all these dogs start out sort of yellow, and then they either produce pigment and become chocolate lab or black lab. I don't know what this guy is. I searched labs and found this picture, and it had like this fourth option, which I was just not prepared for, but I included it because it's a cute picture. So I'll look into that. But I'm only going to talk about three types. The yellow lab has... if we look at this Punnett square here, this dihybrid, so they have big B or they either have two big B's or they have a big B and a little b, or they could have, two little b's. So let me start over with my explanation of this. If a dog has two little b's, he's a chocolate lab. If a dog has one or more big B's, dominant black allele, I guess, then they are a black lab. So the chocolate labs are the recessive, and the black labs are the dominant. Separate from that, though, they have these E genes, which I forget what that stands for. Epistasis? Probably. If they have the dominant form of that gene, then they will express their color gene. If they don't have a dominant functional copy of the E gene, then they won't express the color genes at all and will stay yellow labs. So you notice that here we have, like, this would be a black lab, but it doesn't have the E's. This would be a black lab, this would be a black lab, but it doesn't have the E's. This would be a brown lab or a chocolate lab, but it doesn't have the E's. And so these dogs here still possess the genes coding for being a black lab or a chocolate lab, but they don't possess a copy of the allele that enables them to even show that coloration. So that's epistasis. And, interesting thing, you could then take a yellow lab and breed it with, a black lab or a chocolate lab, and you could get, you'd have to find out, you don't know what the dog is coding for until it has puppies, and then you're like, whoa, you're actually a black lab, but it's just shielded by epistatic effects. So cool example. So these are our different modes of inheritance, and understanding each of these examples or each of these versions of inheritance is important, and I think the examples can help. Try to keep the different sex-linked, influence, limited, and X and activation ones straight. Some of the other ones, I think just remembering an example is helpful to understanding why it's a little bit different. All of these fit under Mendel's sort of understandings of this stuff, but many of them are things that just he didn't have the means or the organisms to identify as sort of being examples of interesting inheritance. But at the end of the day, his law of segregation, law of independent assortment, and all that kind of stuff still applies to all of these. It's just that biology is complicated, and so we have to come up with words to help us to understand those sort of niche examples of things that just seem different than other traits. So there you go. That's a lot of me talking. I hope you enjoyed, and let me know if you have questions.